Welcome to Unit 3 Biology. In this video, we're discussing our first study design dot point about nucleic acids. We need to know that nucleic acids are information molecules that encode instructions for the synthesis of proteins. We need to know about the structure of DNA, the three main forms of RNA, and we need to be able to compare those things. Now just remember that synthesis means to make. So while we don't actually talk about building proteins in this dot point, this is telling us that nucleic acids are very important for building proteins, and it is something that will come up very soon. So nucleic acids are molecules that are found in every living thing that we've found on Earth. Obviously, we don't know about the objects that aren't on Earth, haven't even found any living things just yet. They might have a completely different information storage. We just don't know. But for your knowledge for Year 12, you need to know that nucleic acids are found in every living thing. They store information and they code for cell function. So they tell cells what to do, how to do it, when to do it. Our nucleic acids are the instruction manual for our cells. We have two main types, DNA and RNA, and you do need to know about both of those for your study design dot points. Now, we have already learnt a little bit about DNA in Year 11 biology. We talk about DNA replication, and we talk about it in the context of genetics in passing down information. It comes up when we discuss mitosis and meiosis as well. This is a deeper dive into the structure of DNA. Now, DNA stands for deoxyribonucleic acid, and you do need to know that one. It does come up on the exam occasionally where the examiner will ask you to write out what DNA stands for. Now, when it comes to spelling, you should try your best to be able to spell it correctly. However, as long as you're really close and the person marking your exam can figure out what it says, you're okay, you'll still get the mark. So for example, if you swap the E and the I here, that would be enough to get a mark. If you had something completely off base and it says deoxyribonucleic acetone, you're definitely not getting a mark there. Now, the really great thing about biology is that we can actually use this part of our word to figure out the structure. What it tells us is that our DNA contains ribose, which is a type of sugar, and the deoxy tells us that there's no oxygen attached to that. So we know that somewhere within our strand of DNA there will be a sugar and there's no oxygen as a part of that sugar. So it's a deoxyribose. DNA is found in the nucleus of eukaryotic cells. So if we remember back to Year 11, it was the very first thing, it's back in Unit 1. A eukaryotic cell is a bit more of a complex cell which has membrane-bound organelles. So it'll have a nucleus, which is where you'll find all your DNA. They'll have mitochondria. They'll have some Golgi apparatus. They might have some endoplasmic reticulum, either rough or smooth. And eukaryotic cells can also have ribosomes, and there are other organelles as well. So our DNA is found in the nucleus. It doesn't leave the nucleus of a eukaryotic cell. This is really important to know for later when we talk about transcription and translation. Prokaryotic cells are a little bit different. Again, we remember back to year 11, our prokaryotic cells tend to be a lot more simple. They'll have that cell membrane, they'll have some free-floating ribosomes, and then apart from that, they don't really have any organelles. So the DNA in prokaryotic cells is just going to be free-floating. Sometimes the DNA will even look like this, like little circles. This is really important. Those little circles are called plasmids, so little circles of DNA, and they do come up later in year 12 biology. So this DNA is encoding all of the instructions for that organism. Doesn't matter if it's a bacteria, a fly, a horse, a fish, their DNA codes for all of those instructions. If we looked in a human cell, we would find about 3.2 billion base pairs. And our base pairs are this here, that sort of vertical alignment. So we would count 3.2 billion this way, but then they're paired up like this, so we don't need to count back this way for 6.4, we just call it 3.2. That's a lot of DNA. When we're talking about the structure of DNA, we say that it's double-stranded. So we can see one strand and two strands. We say that DNA is anti-parallel. We can see that in that smaller image that featured on the previous slide. What that means, you would have noticed these numbers, 5' and 3'. And you can see that the strands are sort of opposite each other. Our 5 matches with a 3 matches with a 5. So that means anti-parallel. The strands are still parallel to each other, but they run in opposite directions. We go from 5 to 3. So they're right next to each other, but they're read in opposite ways. You don't need to know too much more about the theory behind the anti-parallel. You just need to know that it's a thing. That will come up if you pursue this at university. When we look at DNA, our backbone, so the backbone is this bit of the strands, is made up of sugar and phosphate. And those two substances sort of alternate. You'll have a sugar attached to a phosphate, attached to a sugar, and so on, all the way up. We also find within the structure of our DNA 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. And we commonly shorten those to A, T, G, and C. And what happens here is that those nitrogenous bases are complementary. So they have another base that they will always pair with on that opposite strand. A will always pair with T, and G will always pair with C. And so a little mnemonic that my students have used before is that they will remember those pairings by saying, at the Gold Coast, A and T, G and C. Now, you might remember that we also talk about chromosomes and genes. Comes up in year 11, comes up again in year 12. So we say that DNA is the building block of this. A chromosome is just a really long strand of DNA and some proteins in there, not important for year 12. A subsection of that chromosome is a gene. That gene is also made of DNA. So we can add to this little image here. The chromosome is the whole structure. If we just looked at one section, it's called a gene. And that gene and the whole chromosome is made of DNA. You could think about it this way. The whole chromosome is the instruction manual. And in humans, there's a lot of instruction manuals. We've got a lot of chromosomes. But just think about one chromosome being one instruction book. The letters within the book are your nucleic acids. It's that sequence of bases, A, T, T, G, C, G, whatever. A gene is just a chapter of that book. If you're reading a book about your washing machine, that book itself would be the chromosome. You might want to jump straight to the chapter about troubleshooting. So that would be a gene. And every single letter that you read within those words would be your nucleic acids or your nitrogenous bases. Now this is an image from a past exam question. This is a nucleic acid, and it asked for students to label the 3 parts. So what we have here is our nitrogenous base. You don't need to provide an example. You don't need to say that it's A or G. You don't have that information. You just say that this part here is the nitrogenous base. Over here, this small part is our phosphate. And if we're talking about DNA, this section here is our deoxyribose. Now, as it happens, this question actually came up in the context of RNA, so it was a little bit different, but it still applies to DNA. It's really important, and this was noted in the examiner's guide. This is not to be referred to as a sugar on the exam. It doesn't provide enough information. Students needed to call it deoxyribose. Or in the context of the question, what it was. We'll discuss that later. Now we also need to know about RNA. And RNA stands for ribonucleic acid. So you would have noticed already that the key difference here is that it doesn't say deoxy. Now that implies that there is oxygen involved with this ribose, with this sugar. You don't need to know about where it is. You don't need to know the specific structure. You just need to know that the type of sugar is different. Now, RNA is actually found in a range of locations in the cell, and that's because it comes down to those 3 different types that were noted in that study design dot point. We have mRNA, tRNA, and rRNA. So we can find those different types of RNA in different parts of the cell. We will discuss that. Now, there are some living things out there that only have RNA. For example, some viruses will only carry RNA. There's not really much else out of that, but that's good to know. Now, RNA is single-stranded. It only has that one strand or that one backbone. And RNA has 4 bases as well. Most of them are the same. We have adenine again, guanine and cytosine. But really important to note that RNA has uracil, not thymine. So that's one of the key differences between DNA and RNA. Now, even though RNA only has 1 strand, it will go and pair up with other things while it's fulfilling its purpose. We talk about that later in the unit. So you do still need to know that A pairs with U, as our U has replaced the T, and G pairs with C still. So when you think about RNA, just remember that instead of thymine, we don't have that. we have uracil. And T and U are right next to each other, so just keep remembering that they're a replacement. Now, mRNA is messenger RNA. You would have heard a lot about this in recent years with the development of the COVID vaccine that uses mRNA. It's really important for making proteins in cells. Super important. We tend to learn about protein synthesis in the context of eukaryotic cells, which is reflected in our summary here. We say that mRNA starts in the nucleus and then moves to the ribosome. And we actually learn a lot more about that later. At this stage, you just need to know that its location is in the nucleus and then the ribosome. It's still single-stranded. It still has A, U, G, and C. And it's needed for protein synthesis. And as a matter of fact, our tRNA and rRNA are also really important for protein synthesis. tRNA stands for transfer RNA. We find it in the cytosol, so just in the cell, and it moves between there and the ribosomes. It has a specific shape that you need to know the general gist of, and it tends to look like this. So that little bump there, some sort of bulge at the bottom, another little bump coming out, and then normally a circle at the top. Whenever you see something like this on the exam, you know they're referring to tRNA. Again, we talk about this more in a later dot point. Finally, we have rRNA, or ribosomal RNA. All you really need to know about this one for Year 12 is that it's what ribosomes are made of. We don't need to get into too much more detail about that one. So to summarize, we have mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. Now, we do need to be able to compare these. So these are the most common distinctions that you need to know. DNA has 2 strands, but RNA has 1. So we can say that DNA is double-stranded and RNA is single-stranded. We do need to know that DNA has that deoxyribose and RNA just has ribose. We need to know the bases, and luckily 3 of them are the same. We have adenine for both, guanine for both, cytosine for both, and our distinction, DNA has thymine, RNA has uracil. Normally in an exam question, if it asks for the distinction, you would just say DNA has thymine as a base, RNA does not have thymine, it has uracil. It does depend on the wording of the question though. And finally, this really should say location in a eukaryotic cell, if we're talking about a nucleus. DNA is found in the nucleus. We don't find it anywhere else. RNA can be found in different locations in the cell, depending on the type of RNA. So in the study design dot point, you need to be able to compare DNA and RNA. That means that you need to know each one in enough detail that you can highlight the differences between them. Again, this is an image from a past exam. It might just say identify the types of molecules here. In molecule 2, we can identify that there are 2 strands, which tells us straight away that it's a strand of DNA. Molecule 1 has 1 strand, sort of. That's how we refer to it. It's also got that shape that we pointed out previously. So we know that it's a type of RNA, but specifically looking at the shape, looking at the fact that you can almost draw on the top of it and see we've got down and across, it makes a T. So we know that this is tRNA or transfer RNA. That's all for our types of nucleic acids. I hope that makes sense. Always refer back to your study design dot point. Make sure you're practicing your exam questions. Thank you.